Right to go? Good. So I should introduce myself first. I'm Rob Dunn, Director of Discovery and Preclinical Research at DNDi. And what I'd like to talk about briefly is how we've evolved in discovery research at DNDi, our position on IP and how that fits and how we want to evolve to initiatives such as open source. And then for me, some questions and concerns that I think my colleagues at DNDi have in terms of how open source would work for us and what we see as some of the issues which may be best. So firstly, DNDi, we're a PDP, much like NMB, a little similarity there. We're a drug development PDP, going from basic research through to product on the market. We now have six products with access to patients. Our portfolio is really the most neglected diseases, and the list there is I want to go through. We have 7. The last, malaria, is in transition to MMB, and I think by this year it will be part of the MMB portfolio. If I have the next slide, I'll talk a little bit about how we've evolved and how we're looking forward. So in the early years of DNDi, we mainly focused on academic alliances for a couple of reasons, but one, I think we evolved from Medicine Sans Frontières. So most pharma companies have a healthy willingness of dealing with this. But we found over the first few years that for the diseases we were working on at least, there wasn't very significant funding for the very, very neglected diseases. So there was a very significant lack of critical mass in the research groups that we worked with. And for us, it would have been extremely expensive to build some of those into functional, I suppose, drug discovery groups. And there was low flexibility. Most of those groups were focused on a particular target or a particular type of chemistry. And should that fail, they were very, very unlikely to change the research strategy. So after a few years, we moved more to a virtual biotech model, I would say, with a focus on collaborations with pharma. And for us, the short-term goal has been candidates in the pipeline and in the clinic, because I strongly feel that PDPs are an experiment. We're 10 years old now, it's our 10th anniversary. We've shown in the last 10, 15 years that we can do clinical research and we can do development in very, very difficult environments. But I think if we don't show that we can get drugs into the pipeline and to patients, then the concept of drug discovery in PDPs will be regarded as a failed exercise and the funding will dry up. So I must admit their primary goal has been to get those drugs through whatever mechanism, and to date it's largely been, I suppose, pre-competitive research, working with predominantly pharma in areas of areas such as data mining, looking at diversity, accessing their technology platforms and so forth. It is pre-competitive. We can't, at the moment, for many of the projects, put that in open access because we are bound by certain agreements with pharma that we do look to make the final product open access, as I'll show on the next slide. But looking forward, we will maintain the virtual biotech model. We will continue to work that way, but we want to now explore more sustainable models. And the last point is that Rob Dunn pointed out DNDi. I don't know if that's DNDi's philosophy, but I think we should aim for programmed obsolescence of PDPs. We should be facilitators of change to bring in new and innovative mechanisms of doing drug discovery, but I don't think being virtual biotech is the complete answer. So the next slide, this is really looking at our approach to IP, and we see what we want with our products and have done to date is to take those to market as generics. So to take the restarted research, to license them to pharma or generic companies as generic products. And in doing so, we can guarantee a sustainable supply, hopefully competitive manufacture, where there's not many companies who want to compete for the markets that we're in. And with that, no IP constraints. So we're really looking at, and I think we go once again here, trying to absorb the research costs so that we can produce a generic product. And this has worked for us already. So we go to the next slide. So we're looking at obviously many different ways on how we can absorb those research costs, and obviously funding and sustainable funding which matches the development timelines of drug discovery and drug development rather than shorter term funding that's mainly available. Non-competitive research strategies, pre-competitive and such as public-private collaborations. Increased access to intellectual property and IC, which I call intellectual capital, which I think is probably more important than intellectual property, which is the know-how of the people behind the patents and the IP. at new models such as open source and how we can progress with open source. I won't go into development. I think obviously there again we'll need large public funding, and I think as Rob mentioned, I think drug resistance, most common infections are going to be a big test case in coming years for that. So I go to the next one. So these are a couple of concerns now, and the two concerns I have are incentives to bring people into open source research. I think it's been discussed previously as well. And the second is the process itself of drug discovery, and the concerns I have there. So the incentives, of course, I think open source will predominantly be done in the academic or the public sector. I think there are too many constraints with many of the industrial companies to commit to an open source process. It's not absolutely impossible, there are constraints. I think the current reward system for public research doesn't favour drug discovery. It still favours academic science, and that is hypothesis-driven science, as opposed to product-driven science. And I've looked at a number of, not such open source, but academic groups doing very good research in drug discovery, but very often they come to the realization that they do have to face that risk of massive failure after several years. And that is a major concern, and leads to what I consider to be often salami science, where a big project will be cut up into many, many little slices, looking at many different funding opportunities, and the vision of that end product is lost. And I put down there a couple of things that I thought may be things we could discuss in terms of incentive. Publication is obviously one. One that came to me recently, and maybe it'll be out there as well, is collective postgrad degrees, because you need a range of different disciplines in drug discovery. And the possibility of having a group of postgraduates rather than each doing its individual project, and that group covering the breadth of disciplines required for drug discovery may be a good incentive in terms of getting a postgraduate degree. And then I think the incentives in the public sector, such as judging tenure and funding by people who are drug discoverers, as opposed to the more academic approach to judging at the moment. And finally, one of the important things I think is the driver. It's nice to be working on drug discovery for neglected diseases, but if you don't honestly believe that process will get to the patients, it sort of dries up. It's hard to maintain that incentive. So really a guaranteed path to patients. So looking at the same time as you're looking at discovery, at drug development, how that will be done. I think PDPs are one opportunity, but other ways of doing drug development to guarantee that patient access. And the last slide is more on process, and through concerns I have on the process of drug discovery. I was going to say medicinal chemists have short attention spans, but Charlie, who's head of drug discovery, is a medicinal chemist patient, but it is one where you need a rapid turnaround of information. Also, they hate error bars, and I think that's important, particularly in biology. It has to be validated biology. It has to be systems which are reliable. We find even working with single laboratories that we get quite a degree of variation when we work with a couple with different strains, etc. It's really quite variable. And the other is, again, talking about this management and leadership, is that you can ask a very, very good question with just a handful of compounds in medicinal chemistry, if each one is thoughtfully designed. On the other hand, you can make 100 random analogues, nudging those well, and it leads nowhere. You don't answer, you just ask more questions. So the final thing there, I think, is the chemistry. We need, with chemistry, leadership to give well-defined questions on what the chemistry will do. We need efficient and validated biology, but not only that, biology which is tailored to the question that's being asked, and not simply a throughput in a couple of assays which are routinely used, but sometimes to modify those assays. And a rapid turnaround to keep that process going, to keep the enthusiasm. But also, I think, in terms of timelines, it takes, 2 years in a good optimization program with a very, very strong focus in a team. If you've got that out without that focus, without the turnaround, I think it could go to an extent where it loses interest. So these are questions, I think, and I'll leave it at that, and I'm sure there'll be some discussion later on. So thank you.
Unless anyone's got a very quick comment or question. Short persevere. Lewis, you want to?
Yeah, sure. Can I ask a quick, you're waiting. When the generic companies come to you, what sort of things, if any, do they ask you in terms of their needs?
Well, we've actually worked both with, it's been a collaboration, so it's not them coming to us, we certainly have to approach them as potential partners to distribute. As I say, we have a couple of products which have worked as generics. I think in our market it's not such an issue because there is really no value in the market for the neglected diseases we're looking at most of them. So I suppose, echoing a lot of the kind of think the issue, but it really comes down to negotiation with them to make sure that they do get some value rather than the practical loss. And that's very important to us too. We think that donations, if people want to donate or companies want to donate, that's wonderful, but it is not a sustainable mechanism for keeping drugs out there with patients. So it's really a cost plus small profit that we're looking at in terms of the negotiation there.
So there does need to be a value chain.
Yeah. Well, we would prefer that in terms of making sure that it is sustainable.
Okay, great, thank you.
So, well, first of all, thank you to Matt for inviting me. I think it's great to have and see this community of people that is I don't think we meet together often enough. There's not that many and a big job to do, so it's great to be here. So if you move on to the next slide, please. I'll first describe a bit our center in Tres Cantos. So Tres Cantos is north of Madrid. And we have two units, the disease of the developing world. This is a end-to-end drug discovery unit that takes care of all clinical development. And then the CIB, and that's a very happy coincidence with us when there was a merger between Barcelona and Midline region. This was the GW side and this was the Midline side. So this was the screening center, and this was great news for us because it's one of the corporate GSK high-throughput screening centers. So we have the capacity to take things from the very early stages of drug discovery up to candidate selection, and now actually through support in steerage, we also have the capacity to do clinical development. So if you move on to the next slide, please. Our model is different in each one of the disease areas. There's commonalities, but it's not exactly the same. So in malaria, we work together particularly with the MND, and we have a large portfolio with significant funding. There we have close ties between each other, and we drive projects together. In the TB DPU, we have also the same type of arrangement with the TB Alliance, but this alliance is a bit more modest inside. So we have complemented this through working particularly with academic collaborators through funding from different sources. We have the MIND scheme, the FP7, Gates Foundation funding, etc. And the Geneva DPU is more kind of a virtual unit. There's only a very limited internal effort, and then we have and rely on more external partners, academics, through the Scandal Product Foundation that I'll go into more detail later on. An alliance with DNDi that is crucial for us as well, and more recently some Wellcome Trust funding and the collaboration with the drug discovery unit in Geneva. And what's very important to mention is that we are fully integrated with GSK units, so we have access to molecular discovery research for screening centers for the MPK for progression of compounds, animal sciences, and also the clinical units that I mentioned earlier on. So if you move on to the next slide, please. What I've tried to look here is to compare this open source concept. So when I started working on drug discovery in TB about 10 years ago, I struggled with, I have to say, people were saying open source, and I couldn't see it. I just couldn't see it. Why? Because differences for me from the drug discovery background were massive. And ignoring it, I think, is a very, very major mistake. So what does drug discovery need? We need big space, we need facilities, we need equipment, and those cost a lot of money. Software development, the open source, you have yourself, your computer, connected to people, easy. Regulation, this cannot be ignored. This regulation is there to protect patients, and you can imagine, you know many examples where this is very, very important. There's very little regulation. The free software concept basically is based on that. The cost, probably we would talk about orders of magnitude difference between one and the other. And timelines. So drug discovery project, 10 years minimum, and software development is something that can happen within So when we thought about what GSK could do to address these differences, we came up with 3 principles. If you move on to the next slide, please. What could we do? We could provide access to our know-how, facilities and funding. That was very important. You need money to push the whole thing forward. We have facilities that I'm not going to say that they are underused, but by giving access to third parties to them, maybe we could make the best of them. And the know-how. This unit has been there for 20 years and we have access to the rest of the GSK expertise. So if you brought drug discovery projects in, maybe a combination of external and internal knowledge would be able to drive those projects forward more effectively. And how have we done that? We're setting up all the standards of our foundation, and I'll describe this initiative more later on. We have also engaged the full 97 program, hopefully the Horizon 2020, successfully by the creation of some large consortia trying to combine the best of two worlds, from the academic and industry. And I think that we were one of the first farmers to actually coordinate one of these big cooperation programs. And more recently, we have reached, or we are having very good understanding with the Wellcome Trust, where the Wellcome Trust believes in the way we are pushing things forward, this open collaborative model, and they are supporting us enthusiastically. Second thing that we thought that we could do is this sharing of data, so that DNDi has talked a lot about that. We are big contributors to the Mary Box, and actually we have also done the same thing in TV, through a more release, recent release of TV data in Chem, and we hope that there will be a second generation of compound antimicrobial and malaria data very recently. And through the PDPs and more particularly through WIPO, we are trying to find an intellectual property framework that allows us to embrace the concept of open source, but at the same time respect the internal tensions that do exist in pharma. So if you are an academic, you know, that can come here and very freely say those principles, and nobody will jump on his, or maybe some people in his university, the next transfer office maybe won't be very happy about that. But we should find balance, because if you want to engage industry, you have to realize of the beliefs that exist there, and if you want to change them, you have to first understand them and change them very sequentially, and not in big kind of dramatic transformations. So if you move on to the next slide, the Open Lab. What is the Open Lab? The Open Lab actually is a foundation that is independent from GSK, receives a significant amount of funding from GSK, but also from other parties. And what is it there for? So the idea is there's a lot of expertise of basic biology, exciting science outside in the academic environment particularly, but maybe these groups need the pharmaceutical infrastructure for their hypothesis to be tested, and if they are successful, we move forward and hopefully into candidates quickly. So this foundation was created about 3 years ago, and the idea was to open calls for external groups to come to Tres Cantos and work with us on their drug discovery projects. So our motto was, we don't care where projects come from or where they go to, as long as we can catalyze and speed up the progression through the drug discovery pipeline. So I would say that this has been successful, even more successful probably than some people at GSK internally thought. It's been hard, because imagine adapting all the internal mechanisms to having external scientists that bring their projects in and actually take their intellectual property out. So there has been some tension internally, but I think that we have handled them well. So we have over 150 projects. We have presently 50 projects active in Tres Cantos, and there's 3 calls a year. So if you know of any academic or people in biotech that have international projects and need the funding and the expertise, either the screening facilities, the MPK, medicinal chemistry, we are there to help. So you move on to the next slide. Second principle, the sharing of internal compound data sets and data. I mentioned the key counts, the chunk went into the malaria box. Also this more recent publication of the PD data set. All this is available through MDL, and they are very good partners of us. And this the malaria box, as we mentioned earlier, I think it's 7 months since we released the PD data set, and it's been already shared with more than 20 groups, and we have conversations with the MLB for these compounds to become part of the pathogen box in the future. And there's again some tensions because antibacterials internally sees the movement, and eventually the discovery and development of antibiotics will have to embrace this kind of setup, but there still remains of a more traditional view of protection of intellectual property. But I think we'll get there, it's just a matter of time. So if you move on to the next slide, sharing of IP, I think there's people here who are much more qualified than me to describe how this worked. We started up with Hope for Open Innovation, but then evolved in these WIPO Research principles. But I think, when, in a nutshell, what I have to tell researchers what they are getting involved with when they go to the open lab, I think those are the two key things. So patents still work for us, but those are the two conditions that we should embrace. So include, there will be anything that comes out from this research should be royalty-free for these neglected diseases, and be royalty-free for sales in the world in these developed countries. And I think, again, this is a balance between the two tensions where medicines need to be developed for people that can't afford them, but that we respect the intellectual property framework that is out there. Maybe it's not a job for us to change it, maybe it's a job more for the alliances or other organizations to change this framework. And I think that just a very brief introduction to what we do. We will talk many more things, but I try to focus on things that were relevant to the discussion that we will have today.
Thank you so much. I should mention also that the Open Source Malaria project with MMD comes from your TCAN set. So in that sense, the provision of the public data did provide a catalyst for that project because you have a realistic starting point, which is high quality. Because of the quality that you were able to, we were able to generate funding for that project because the review is so, wow, it's really great content. This is obviously going to be a decent project. And also when you were talking about expertise, it's also the granularity that's very important. So during the project, occasionally we have questions about the compounds we make. Are the compounds we're making novel? Can they be sourced anywhere else? Are new compounds, are they already known, have been tested and found to be inactive? And the team at Contest have been very good about dealing with those little granular questions every now and again to help speed things on. And it's that kind of, it's the personal input, which will be so important.
For us, the philosophy is, if a project comes in, either interest cancels or we get engaged with it, we should treat it as any other project. That's what value that we bring on the table.
But it's a cost.
It is cost, and it's people's time, and that's attention internally as well, because they say, you have to be careful that you don't get distracted from your own internal pipeline. But I think the agencies are valuing that.
Okay. Great, thank you. We should probably move on. Thank you so much. And so from industry to the academic perspective, Chad Banter from SGC in Oxford. Is there any transition or is that coming?
I'm so sorry, it's just that microphone's run out of battery, so I'm relying on a directional microphone.
The camera is better.
I can do, but
It's going to be these guys,
it's okay. We'll make lights turn around. It's fine. Cameras
I
won't
think
get
you'll be alright then.
Yeah, it's okay, I'll
No,
move
it's
it.
okay, it's just we're on the microphone.
Well, thank you very much. Good morning, everybody. So what I'll do is I'll talk a little bit about the SGC, what it is, what impact we've had, and what our future plans are. That's the next one. One of my biggest concerns is that in biomedical research, this is basic research, but also in drug discovery, there is massive duplication. So this is an example. So you're all aware in humans there's one family of proteins called kinases. In humans, there are 518 kinases, annotated here, 1 down to 518. This just plots how many publications there are on each of those kinases. And you can see that 90% of the publications are on about 50 kinases. There's 300 kinases that we've not even touched. But of course, if we want truly novel medicines, we should be targeting novel kinases. If you look at the patents, not surprisingly, the patents are on the kinases that are published. There is massive duplication. There is not enough innovation in drug discovery. Could I have the next slide? So what do? So we only work on novel proteins. I'm not interested in working on proteins that other people are working on. We're 200 scientists, about 100 in Oxford and 100 in Toronto. What we do is, for those proteins, we generate new tools. So the protein itself, assays, inhibitors, antibodies, structures. And all these tools, we give them away. We publish them immediately, and we give them away to anybody who wants them, in academia, in biotech, in pharma, etc. Because we believe that's the best thing we can do to facilitate science and therefore facilitate drug discovery. Go to the next slide. So we've now become a hub for innovation. We only work on new proteins. We've also become a hub for pharma. We've now got 8, and soon we'll announce 9, pharmaceutical companies working with us, and each of them have contributed $8 million to our effort. So we have $72 million of private funding. The companies are GSK, Pfizer, Novartis, Lilly, AbbVie, Boehringer Ingelheim, Takeda, and Janssen, and we'll announce the 9th one very soon. You can imagine, because we generate new tools and we give them away, every academic who comes into my office wants to collaborate with us. They know I've got no secrets, I share all of our know-how, all of our reagents, etc. And that transparency creates a lot of trust, which is brilliant for collaboration, it's brilliant for science, and it's brilliant for drug discovery. We'll have the next slide. This is an example of some of the impact we've achieved. So if we just think of the structures of these kinases that I talked about earlier on, this is a cumulative plot of how many structures have been done. This is time along here. The red line is structures that have been done in academia, the whole of academia apart from us. The green line is all the structures that have been done in industry. And this blue line is the structures that have been done by one of my groups in Oxford. So one group in Oxford has now done as many structures as the rest of academia put together. This is the impact they're having. Next slide. And membrane protein structures. So these are proteins that are in the membrane. People are getting Nobel Prizes for these. These are phenomenally difficult to do. 3 years ago, nobody in Oxford had ever done a human membrane protein structure. Let me share with you an example. Last year we did this one, in May last year. In summer, and we published all of the data. This is how we did it, these are the coordinates, etc., etc. In summer, another lab in the US used our data to get the yeast structure. So I only work on human proteins, we did the human, they got the yeast. We submitted our paper in November, and it got published this year, both of them together in Science. So although we disclosed everything to the world, we helped another lab do science, and we still got a high impact paper. Next slide. We've now started to generate inhibitors for these proteins. So this is a novel protein family called bromodomains. We generated this inhibitor, and this inhibitor just hits these proteins up here. You can see it has little or no activity down here. This is a protein family that nobody had ever worked on before. This is the first inhibitor for this family. We're opening up new chemistry. Next slide, please. We then gave that inhibitor, that same inhibitor, to a clinician in Harvard, and he put it into a cancer model, a rare type of cancer called nut midline carcinoma. And what he showed was that in an animal model, if you didn't give the compound, I'm sorry this is a bit out of focus, but in the presence of vehicle, this is a cancer, the tumor growing, and if you give the compound, you can see it stops the tumor growing. At this point, Jay Bradner in Harvard wanted to take that molecule into patients because anybody who's diagnosed with nut midline carcinoma is dead within 6 months. There is nothing out there that works. Next slide. This has been the impact of that one inhibitor. So we published it in Nature in December 2010. By May this year, that paper had been cited more than 200 times. By May this year, there had been more than 170 papers as a direct consequence of that one paper. We've now given that molecule out to more than 300 labs all over the world, and they're looking at it in other types of cancer, inflammation, infection. You can imagine the breadth and depth of science that's being generated by that one inhibitor. Many of our pharma partners have started proprietary programs on that target. And Jay Bradner secured $15 million of VC funding to set up a biotech in Boston. This has been the impact of one freely available inhibitor for a novel protein on science and on drug discovery. Next slide. Drug discovery at the moment, I think, is a complete lottery. I'm focused on drugs for Alzheimer's and diabetes and cancer, etc., etc. It is a complete lottery. Most novel targets, pioneer targets, when we take them into patients for the first time, so phase 2, the failure rate is more than 90%. More than 9 times out of 10 they fail. We as a biomedical community are not good at target validation. We are not good at saying this new protein, if I inhibit it, it's going to work in this patient group or this patient subset. This is why the industry is in such a crisis. Target validation only occurs in the clinic. It's only in patients we really find out if a novel target is going to work. It doesn't occur in animal models, and I'm afraid it doesn't occur in cells. Unfortunately, many academic groups and many companies, they are working on the same few targets. So now imagine, we've got 20 companies working on the same target, in parallel, in secret. They'll generate a proprietary molecule. It'll take them 4, 5, 6 years, and when they go into patients for the first time, 9 times out of 10 it will fail. What a horrendous waste of money, of people's careers, and patience. The way we're doing drug discovery today, we are exposing patients to molecules that other groups already know are destined for failure. It is unethical. Next slide, please. So what we're now proposing to do is to set up another public-private partnership, a bit like the SGC. Now we're not just going to generate reagents and give them away. We're going to generate clinical candidates, and we're going to take those clinical candidates all the way to patients, completely in the absence of IP. This is a knowledge creation endeavor. The reason drug discovery is a lottery is because we do not understand human disease. This way, if we don't take out IP, make stuff freely available, we can access the best academics quickly and freely. The targets we work on will be selected by industry. The patient groups and regulators have said that they want to be active participants in this endeavor. And industry, once we've identified that 1 in 10 that actually does something, then industry will generate proprietary assets. At least then they know they're working on clinically validated and de-risked targets. Next slide. So what we're intending to do is to literally go through all the way through the pipeline to the end of phase to do some proof of clinical mechanism study. In the process, we will generate these reagents in blue. We will generate these publications in red. All these reagents and all these publications will facilitate more collaboration. They'll allow us to leverage more funds, and they'll frankly allow us to do more clinical studies. Next slide, please. What's the status of this project? We've already started it in cancer with Cancer Research UK. There's the target, there's the patient group we're thinking of at the moment. We're about to start it in neuropsychiatry. The Canadian Institute for Health Research hosted a meeting a few weeks ago in Ottawa with several heads of R&D and CNS heads from large pharma companies. They're going to put $30 million into the pot to kick this off. We will get other public and philanthropic funds to go with it, and currently we're sorting out what disease we're going to work on and what targets. And we're also having discussions with the Helmsley Trust about doing the same in inflammatory bowel disease. This is very early onset inflammatory bowel disease. Next slide. So final slide. What we're trying to do is to create a more efficient ecosystem. The current ecosystem for drug discovery is inefficient, it's wasteful, it's duplicative, and we're harming patients. We're trying to combine the strengths of different stakeholders, bring together academics, with industry, with patient groups, with regulators, with public funders, etc., etc. We're accelerating innovation. We're now starting to catalyze proprietary efforts. We've also started to create some new companies. Many of our academic collaborators are taking our reagents and some of the new biology that our academic networks generated to create new biotechs. We're reducing duplication, wastage, and patient harm. Frankly, ladies and gentlemen, I think this is the only way we're going to come up with a drug for Alzheimer's and depression and schizophrenia, etc. Many of you are probably aware the industry, the biomedical community, has been working on Alzheimer's. Let me share with you one target they've been working on. This is beta amyloid. 29 years we've been working on that target. Collectively, we've spent more than $30 billion, and we're still no closer to the truth. We cannot carry on like this. This one disease, Alzheimer's, is going to financially cripple many societies if we don't get our act together. Thank you very much.
Amongst other things, two great examples about economic value of open research, but also things that people were mentioning before about difficulty, perceived difficulty of publishing open work, open source and public domain. Clearly demonstrating not the case, that there are no barriers. You can get high-impact publications with stuff that's already there.
I mean, it's worth saying, so my group, we're about 100 people in Oxford, we produce about one paper every 7 or 8 days.
Yeah. Okay. Excellent. Thank you so much. There's one final talk before we switch to coffee. And this intended to bring, again, a different perspective to all the discussions from Tom Bombell from intellectual property.
Thank you, Matt. Very much.
You don't have slides. If you wouldn't mind just...
Doesn't work. It doesn't work.
Unencumbered. So thank you for inviting me to be here today. For those of you who don't know what the World Intellectual Property Organization is, we are a specialized agency of the United Nations with a specific mandate to work on intellectual property issues and to work on intellectual property, including patents, copyrights, trademarks, industrial designs, etc., in such a way as they support innovation and support development goals. So our Constitution instructs us to support the development of a functioning intellectual property system that delivers benefits to society as a whole. And that, I start with that, that's a very important concept because as several speakers here said, what is intellectual property? Intellectual property is not an end in itself. It is a tool. It is a tool to support innovation. It is a tool to support innovation that delivers things that we want and need as a global human society. So, and in this case here, as we talk about health, obviously it's supporting innovation in the health sector that delivers medical technologies that improve and save our lives. So with that, as a little bit of a theoretical opening statement, I want to thank Matt for inviting us to participate in this and to say, I'm going to stay away from some of the theoretical questions about what is the best model for drug discovery in the 21st century, open source, open innovation, closed innovation. I should also say I've been with WIPO for 4 years now, but before that I worked 20 years in the pharmaceutical industry, including for Merck, the American company, where I very much engaged on the issues of innovation, intellectual property, and access to medicine. So this is my policy background. And bringing this sort of policy background to these questions, but also very much, I have to say, I suppose, a corporate focus on what is the issue, how do we engage on it, how do we get something concrete done, and staying away from the philosophical questions. I was brought to WIPO to develop some projects with external stakeholders that would address the question of is innovation, is the IP system working to develop and deliver solutions that benefit all sectors of society, including the poorest countries, poorest populations, most vulnerable. And how could WIPO, which is not a development agency per se, it's certainly not a health organization, how does WIPO engage on these issues, which obviously do have an intellectual property component because aside from talking about health this morning, what we've been talking about is also intellectual property rights. So one of the things that I've learned in my career and in life perhaps is everybody has a job in life. You have to understand what is your niche, what is your role, what can you do to engage on a certain aspect of an issue. So understanding that WIPO understands that its role is to support the development of effective IP systems, not as a goal in and of itself, but rather as a tool to deliver socially desirable results through innovation. We looked at this issue of neglected tropical diseases. Clearly, the intellectual property system, as it has developed in the 20th century, has delivered tremendous positive results and products that have changed the way that the world is, beginning, I would say, in the early 20th century. I feel like sometimes in the world of public health in particular, we all tend to be a little bit negative, which is, I mean, it's a good thing because we have to act. There are huge, huge problems, and there are a lot of, very bad things that happen, frankly. On the other hand, there's been a tremendous amount of progress. Just one of the advantages of going last is you can kind of collect people's comments and respond to them. So, I mean, as Charles said, which is a great presentation, yes, Alzheimer's is a potentially crippling problem that we face, especially in the developed world today. Why? It's not necessarily because Alzheimer's is an intractable disease as it exists right now. It's because people are living long enough to get it. Let's not forget that. That's my only point. Let's keep some perspective on this issue. So while the innovation system, the IP system, has delivered results that have improved human life as we know it, is it perfect? No, of course not. Does it deliver these benefits equally to all sectors of society? No, of course not. So there is a problem. One particular problem that we thought as WIPO we could engage on effectively. We could use our institutional mandate and our organizational competencies in partnership with others to engage on, and that's the field of neglected tropical diseases, malaria and TB, which, as everybody knows here, I won't spend a lot of time on the background with this audience, disproportionately affect the poor and the most vulnerable segments of society in the world's poorest countries. The private sector, which relies on the formal system of intellectual property rights to generate and invest risk capital in the highly risky, wasteful, and inefficient system that they use to try to discover new drugs, is not investing billions of dollars of shareholder capital, as a general rule, in these neglected tropical diseases for a variety of reasons. Yes, certainly there's a lack of economic incentive, but it's also true that the large majority of these diseases are preventable, and the reason that we don't have them in the middle income and the richer countries is not just climate driven. It's a function of public health and access to health care and all those other kinds of things. Nonetheless, there's obviously a very strong need for new and better treatments. So WIPO Research, or WIPO as an organization, including me personally, looked at this issue and said, how can we design some external partnerships that might use some of the developments in the way that both the public and the private sector, which you've heard here in a number of the excellent presentations, open innovation, open source, clearly, and again, with my background in the private sector and the pharma industry, I'd like to just validate what Charles and some of the other speakers said here this morning. Clearly, there's a very strong sense and a recognition inside the pharma companies as well that their business model as it existed and as it delivered positive results for many of the past decades is certainly not working the same way that it did, and some people go so far as to say that their business model has failed. Again, I don't trouble myself too much with the philosophical questions. I'll do that after I retire. But clearly, it's not delivering results the way that it used to. So they're also looking at, and Luis, also presented that very well, open innovation, open source, etc. So how did we put all that together to create a project called WIPO Research, which is an open innovation consortium. So I stress open innovation, not open source. We used our institutional mandate and the desire of our partners, which are now over 70 and include private sector pharmaceutical companies, academic institutions, PDPs, NGOs, DNDi is a member, GSK is a member, who else? So MNV is a member, thank you. To create a system whereby on a platform, on a database, which is maintained by WIPO, providers of intellectual property can advertise the availability of intellectual property assets, including services such as high throughput screening, which is something that's offered by GSK and other pharma companies, to entities that are investing in R&D for neglected tropical diseases, malaria, and TB. And it's structured to use the IP system as a way that allows for the transfer of intellectual property assets and knowledge for this purpose of stimulating investment in innovation in this neglected area. So one of the things I'd like to talk about more in the discussion is I heard a lot of statements about, we're designing a project so that we don't have intellectual property rights so that there's no barrier. Or we have a no patenting policy so that there's no barrier. Well, I want to explore this during the discussion, but it's important to remember that the IP system and patents in particular are specifically created for the purpose of making information public so that others can use it as a spur towards innovation. So that's why there are patents. And then in turn, patents, in return for the inventor making information public and publicly accessible, the inventor gets certain exclusive rights over that invention, and they are supposed to use that. If they have a viable business model, they generate some income, which in turn is reinvested in innovation. So we can go back in discussion to some of the quotes that you put up at the beginning, Matt, because they raise some interesting questions. So what we see and what attracted our partners to our consortium is that using the intellectual property system makes it possible for them to transfer proprietary knowledge, including non-public information, to other parties with security and safety, and thereby reaching these broader goals. So, is this the only way to do things? No. I would also say that where we are today in the 21st century is that there is this crisis in the model of innovation and intellectual property in the field of health. There is no one solution. You see GSK's presentation again. DNDi would not have existed 30 years ago. It exists today because everybody recognizes that to get these results in these areas, we need to have different models. I'm not saying that my model is the only one. I'm saying that mine is one of them in today's landscape. And let me give one example, and this is just coincidence that it happens to be with my former employer, but it's a fairly simple one so I can understand it, that shows how and why this works, because we're talking about intellectual property, not patents, and those two things are confused. So WIPO Research, I like to call it BP. It's beyond patents. Patents are just one segment of intellectual property rights. They're just one legal mechanism. But intellectual property, writ large, is about knowledge. So there are scientists at University of California, San Francisco, who have a theory that statins, the cholesterol-lowering medicines, may have certain properties that are effective against the schistosome parasite. So they're working on this. Through my project, they were able to connect with Merck, which had a big franchise in statins, and so they now have an agreement. The researchers at University of California, San Francisco are actually getting physical samples of certain compounds that Merck owns. But more importantly, they're getting access to all the published and unpublished data and knowledge about Merck's statin drugs as part of their work. So why is this important? Because one of the big problems, and all of you know this much better than I do, is the so-called valley of death. It's not that hard. I mean, it's difficult work, but people can do it with a relatively low financial investment, to do the academic and preclinical work. That's being done all over the world. What the big problem is taking that work and making it druggable. That's the so-called valley of death. If researchers have access and can use all the information that another entity has, it can be a private sector company or somebody else, has that give them access to all the safety and all the quality information that they need, then the only thing they need to do when they get to the point of human clinical trials is prove efficacy. That makes the world for them much, much easier and simpler. Still costly and risky, yes, but less so. So that's just one example. We have 33 collaborations, 28 of which are on our website. So let me pass these around again. I'll show this to you just to underline my point about just taking a very pragmatic approach to some of these problems and saying, what are the results that we want and how are we going to achieve them? So for more information, obviously we have a website, it's www.wiporesearch.org. And then let me conclude with, again, back to the issue of intellectual property and how does this fit into this. We encourage that if an entity works on something under a WIPO research agreement and they discover new knowledge, new information, we encourage patenting. Let me repeat that. We encourage patenting. Encourage. However, royalty-free licensing is required. Patents are encouraged, royalty-free licenses are required. And that's how we deal with the issue of using intellectual property as a tool to deliver the goals that we as a society want and need, and make sure that the IP system is flexible enough and evolves as society evolves in order to address these issues. So thanks for your attention, thanks for inviting me, and I look forward to the discussion.
Great, thank you. Lots of food for thought. Speaking of which, I think we should break for coffee, which I think is out here. We're not necessarily part of this larger group, which is emerged over in the area there, but there's some coffee and things out here. So maybe about a 15-minute break.